So yeah, so it's just me tonight. You guys kind of know the drill. If you have any questions, don't feel free to ask. Don't feel like you gotta keep it bottled up. All right, so I also can't see the chat because there's no one else to read the chat. So if you guys have questions, could you just like say them just because I don't have access to the chat? 1 second. Okay. All right, so we're gonna go over what we basically went over at the beginning of this week. We're gonna go through up until the anaphase metaphase checkpoint, which is the spindle assembly checkpoint. And then I'm sure Friday's session will cover anaphase and most of the stuff that we went over in class today with anaphase A and B and the final parts of M phase. But for today, we're just gonna do a quick recap. You have the phases of the cell cycle. You guys should know what occurs in each phase of the cell cycle, what the cell needs to do before it moves on. So for G1, you need enough nutrients and machinery in order to get to S phase, and you also want that the restriction checkpoint. That's that all or nothing checkpoint that we talked about. If you don't have that checkpoint, then you won't go into S phase. S phase, pretty easy to remember. Synthesis is just DNA replication. There's a checkpoint at the end before you go into G2 because you're going to want to make sure that you have your DNA replicated properly. And then you have your G2 phase, which is after DNA replication. The cell is getting bigger for mitosis, so it can split. And then we're going to talk about M phase today a lot, up until anaphase, because that's where we'll stop for this one. So we're going to start with the control events that occur in S phase. S phase, like I just said, is the synthesis phase. This is where you're having DNA replication. So once the cells are in S phase, the cell wants to prevent the pre-RC from forming again because we only want that to happen once. We don't want replication to occur multiple times because then we'll have a ton of problems, which is one of the questions I'm going to ask you. So CDK plays an important role in almost every phase, but especially here. It suppresses the reassembly of the pre-RC during the same cell cycle, and it does this by phosphorylating the proteins involved in the pre-replication complex. I know some people were wondering about the PRC, but that's basically just the pre-replication complex for the ORC, the CDC6, and the MCM complex, which is basically your helicase. So CDK suppresses their activity by phosphorylating each of them. So it phosphorylates ORC, which prevents CDC6 from binding. So in this diagram here, you can kind of see it happening. So it phosphorylates CDC6, which breaks CDC6 down. It phosphorylates ORC, which prevents CDC6 from binding, but then it also phosphorylates CDC6, so CDC6 gets broken down anyways, so there's nothing to bind. But if it didn't phosphorylate CDC6, if it still phosphorylates ORC, CDC6 can't bind. And then it phosphorylates specific subunits of MCM. It doesn't phosphorylate the entire complex. It only phosphorylates specific subunits that are involved in DNA replication at the time. I don't think she needs you to know the specifics of that, but it phosphorylates the MCM complex, which causes the MCM complex to exit the nucleus. So basically, by phosphorylating these three proteins involved in the pre-RC, the CDK prevents DNA replication from occurring more than once in the S phase. So the question I have, if you guys want to just answer, I think I can see the chat, but I have to open it. What will happen if this controlled event fails and another pre-RC complex forms? What will happen with the cell and the DNA if this whole suppressing event does not work? What do you guys think?
So if I remember correctly, I think that another round of DNA will be made and then the cell will have too much.
Yeah, so that's basically the idea. The problem with the main problem is that the pre-RC will only reform at certain ORCs. It won't reform at every single one. So you're going to get replicated DNA, but it will be incomplete because you won't have full replication of another DNA strand, which you wouldn't want either. You don't want either of those things to happen, but because you'll have too much DNA, but it also won't even make enough DNA because it will just make like fragments of incomplete DNA because it won't reform at every single complex. Does anyone have any questions before we move on? Awesome. So cohesin, this is just a quick little tidbit on cohesin. Cohesin is a protein that holds sister chromatids together. It's important for preventing chromosome separation during spindle assembly because you don't want that separation just yet. We're going to talk a little bit about at the spindle assembly checkpoint when cohesin gets broken down, but its main job is to hold these sister chromatids together. And then this little diagram shows that you don't have to worry about these terms, but if cohesin is prevented from loading during DNA replication in S phase, then the spindle assembly checkpoint doesn't function correctly, which we'll talk about later on in the presentation. But so you can see here you have S phase. With cohesin, you get proper chromosome orientation, but without it, you don't. And then you can end up with mutations like aneuploidy, which is something like you see in patients with Down syndrome. They have triploidy in chromosome 21, but you just get a lot of mutations. You could get a cell that doesn't function properly from this without proper segregation of chromosomes. And then when you do have cohesin up here, you get proper orientation of chromosomes and proper segregation. So you get the correct amount of chromosomes in each cell when the cell splits. So cohesin is important for this proper alignment. So now we have control events in G2. So that basically sums up S phase. You have your replication. You don't want it to happen more than once. And then you have your cohesin that we're going to talk a bit more about. But that's basically S phase. So now we're going to move on to G2. G2 starts with the control event with the activation of M phase CDK. So M phase cyclins are important for M phase to even start. Without M phase CDKs, M phase can't really initiate anything. So M phase cyclins start being transcribed and translated during S phase, and then they increase during G2. And this M phase CDK is important. And then I'll show the diagram in the next slide, but CDK is phosphorylated by both CAC and WEE1. And if you remember from class, CAC phosphorylates CDK at the activating site and WEE1 phosphorylates CDK at the inactivating site. So if you have both of these competing phosphate groups on the CDK, ultimately the CDK is inactive. And this is actually very important because you don't want CDK to be active too quickly because you want the cell to be able to check for any problems before it moves on in the cycle. So this is where you're going to check your DNA or make sure your DNA replicated properly. And then once the cell determines that everything's all good, then you can sequester in CDC25, which is a phosphatase. So remember, phosphatases remove phosphate groups. So it removes the inhibitory phosphate that WEE1 put on CDK. And now you have this mCDK that is active. So when CDK is active, you can enter the M phase, and we're going to see that in the next slides. But the other important part of this event is the positive feedback loop. So once mCDK is activated by CDC25, I know there's a lot of C's, it's kind of confusing, but once it's active, mCDK can phosphorylate and inactivate WEE1. So then this leads to this positive feedback loop, which allows more mCDK to become activated near the end of G2 when you need more of them for M phase. And it does this by inactivating more of WEE1. So the more mCDK you get, the more WEE1 gets inactivated, and then you can enter M phase. This is this diagram. So I don't know if my pointer, I do. So here you have your M cyclin. This M cyclin levels rise during S phase, and then during G2, this is where they bind together with the CDK, the mitotic CDK. That's what mCDK stands for. And then you get WEE1 and CAC both phosphorylating at two different sites. So you have your inhibitory phosphate caused by WEE1 and your activating phosphate caused by CAC. But because you have this inhibitory phosphate, the entire complex is inactive. So so this is important. You want it to be inactive right here because you want to check the cell for cell growth. You want to check DNA or damage because you don't want to move on immediately into the next phase if you don't check. So this is important. But once you're like, okay, we got the green light, everything's good to go, CDC25 comes in, it's a phosphatase, so it removes this phosphate. Phosphate's gone. Now we just have the activating phosphate, and this MCDK complex is active. And then this is when it goes on into M phase. So this is this positive feedback loop. So you have inactive MCDKs where we start here. You get active CDK, which phosphorylates the phosphatase, which makes you just basically, the main thing with this positive feedback loop is more active MCDK. So it makes more active MCDK. So this makes more of this. I know it's kind of confusing, but the more MCDK you get, the more it inactivates WEE1 and phosphorylates phosphatase, so it's active. And this is what is active. So once it phosphorylates active CDC25, CDC25 can get rid of this phosphate from WEE1 more and more. So it's basically just this loop of making more active MCDK, and this happens rapidly at the end of G2 because you're going to be entering M phase, and you want more of those CDKs. So the question I have is, what do you think would happen if CDC25 developed a loss of function mutation and could no longer remove this phosphate, the inactivating phosphate? What do you guys think?
If the inhibitory phosphate can't be removed, then MCDK won't be activated, and because of that, there won't be a positive feedback loop with the inhibitory kinase.
Right. So exactly, the MCDK complex would stay in this inactive form because CDC25 can't remove this inactive phosphate group. The positive feedback loop wouldn't happen, and you wouldn't have enough active MCDK for the M phase, which we're going to talk about, that has an important role in M phase. So it wouldn't be able to do its job in M phase, and ultimately the cell would kind of halt here, and it would get, it just wouldn't be properly set up for M phase. Does anyone have any questions on this? I know it can kind of be a lot. Awesome. Okay. So now we're in M phase. Once MCDK is activated, we're officially in M phase. CDK has to phosphorylate. So this complex, like I was talking about, has to phosphorylate many proteins at the beginning of M phase. So prophase, you have this little diagram here with all the steps that we're going to go through. So back to here, if this CDC25 has a loss of function mutation, can't remove this phosphate, it's inactive, then this complex can't do these next steps, which are important for M phase. So especially prophase at the beginning. So the active CDK complex phosphorylates lamins, condensins, MAPs, and motor proteins. So lamins are important when they get phosphorylated because they break down the nuclear envelope in prophase, which is the first step. And then it phosphorylates condensin, which is also important because condensin is involved in chromosome condensation. I think we go into that in a little more detail later, but I don't remember. She might have changed it this semester, but anyways, and then it also phosphorylates MAPs and motors, which are required for spindle assembly in metaphase and prometaphase. But so that's why it's important that the CDK is activated from this previous step here in order to do these next first steps of M phase. And remember, just a quick side note, that interphase is the steps from G1 to G2. That's interphase, and then the M phase is this small part at the end with prophase, prometaphase, metaphase, anaphase, telophase. And G0 is technically part of interphase, but G0 is that non-dividing stage. Don't worry about that. So an important part of M phase is this sequential phosphorylation that we talked a little bit about in class. This is pretty important and this is why CDK is technically considered the CEO of the cell cycle, because it's important for its job, dictates how other enzyme's function and ultimately affects the entire cell cycle. So before I go through the diagram, I'll just read this side. But so technically it's a priming kinase. So that just basically means that in order for PK1, which is also a kinase, to phosphorylate the substrates it needs to phosphorylate, CDK has to first phosphorylate that same substrate. So if CDK, no, that's actually my question. I'm not going to say that yet. But so this is important because if CDK doesn't do this, then PK1 can't do this. So it can't, basically this priming phosphorylation adds a phosphate group to the substrate. Then now PK1 can become active because this kinase domain of PK1 can phosphorylate the substrate. You don't have to worry about the PBD, but it's just another domain of the PK1 enzyme that just kind of wraps around this phosphate group. But because it detects this phosphate group that CDK put on the substrate, it can now use this to add a phosphate group with its kinase domain. So the question I have is, what would happen if CDK developed a loss of function mutation and could not phosphorylate PK1 substrates?
Priming phosphorylation won't happen, and PK1 won't be able to bind to the substrate.
Yeah. Yep. So yeah, if CDK developed a loss of function mutation, it wouldn't be able to phosphorylate the substrate. And since it doesn't have this phosphate group, then PK1 also wouldn't be able to phosphorylate it because it's in a sequence, it's sequential phosphorylation. So without that PK1, which is an important kinase for the G2M transition and spindle assembly, if PK1 doesn't do this, it doesn't phosphorylate the substrate, then there wouldn't be proper spindle assembly and you wouldn't have proper splitting of the cell during M phase. So does this make sense, this sequential phosphorylation? Do you guys have any questions? No, cool. Okay. No, go away. All right, so spindle assembly. So this occurs in M phase. It starts during prophase. Basically, the cell disassembles the entire original microtubule complex and reassembles the tubulin into a spindle-like structure. This requires those MAPs and motor proteins that I talked about back here, which CDK has to phosphorylate for them to work. So it's all in a sequence, if you've noticed. One thing has to happen for another thing to happen for another thing to happen. There are different types of microtubules, asters, interpolars, and kinetochore microtubules. Kinetochore microtubules, this is not the same as kinetochores, which are something different. An easy way to remember it is that kinetochore microtubules bind to the kinetochores on the sister chromatids. So this would be the kinetochore microtubule binding to this kinetochore protein complex, which is about 80 proteins. I don't know if you remember, but she showed that diagram in class. It was pretty complex, but kinetochores have a lot of functions in the cell. But the main thing to know here for spindle assembly is that the kinetochores are the binding spot for kinetochore microtubules. And then this is the diagram we showed of different types of microtubules. So the aster microtubules on the outer edges of the cell, they connect to the cell to an area near the cell membrane, not the cell membrane, but it's near it. That's these green ones, they out of the centrioles. Your interpolar microtubules are these pink ones here. They overlap near the center, which we'll talk about in a little bit, the importance of that. And then you have your kinetochore microtubules, which are these blue ones that attach to the kinetochores of the sister chromatids. So that's basically the three types of microtubules you should know. They all have important functions of their own, but the main thing for spindle assembly is that they each are in different locations, they bind to different parts of the cell, and that leads to stability. And this is important, this overlapping here of the interpolar microtubules, which we're going to talk about actually in a second. So they're important for the building of the spindle, so now we're in metaphase, we've left prophase, we're now in metaphase. And so that's this push-pull diagram, which you can kind of see here. These are these motor proteins, one's going one way, one's going the other. And then this is more of just a diagram of what's happening. It's kind of just like a simplified version of the one on the last slide. You have your astral microtubules, kinetochore microtubules. They don't really show the interpolar microtubules here, but so the push-pull mechanism is that basically the theory is that opposing forces keep the spindle stable. So this pushing and pulling, they basically keep the spindle poles a particular distance away from each other so that they're stable enough. And they also contribute to the forces required to pull the chromosomes apart during anaphase. So basically how this works is the minus N directed motors, and quickly, can someone tell me what is a minus N directed motor?
Wasn't it
based
called
on
like
what
Dicene or something?
Dynein, yep.
Dining.
Yep, so Dynein is a minus N directed motor. So this is important. They bind to aster microtubules, so the ones out here but near the cell membrane. They start at the end of the spindles and they move towards the minus end, which creates that pulling mechanism. So the minus N directed motors pull away from the cell membrane towards the center because they start walking towards the minus end. Or sorry, not towards the center. They start walking to the minus end, which creates that pulling. And then the plus N directed motors. So this time it's EG5. I know kinesin's a big one. I actually don't know if kinesin contributes to this. I don't remember if she said that in class or not, but I know the main focus of this motor protein is EG5, which is a plus N directed motor. So plus N directed motors sit on interpolar microtubules. So right in this diagram, this is where you would find your EG5s in these centers here. And they sit on that overlapping portion of the interpolar microtubules, and they start in the middle and then walk towards the plus end, which brings the poles closer together. I wrote inward pulling force because that's technically what it is, but ultimately this is the push that you get from the push-pull mechanism. So the main takeaways from here is that you have your minus N directed motors, so Dynein, which bind on your aster microtubule. So they start on the outside. Plus N directed motors sit on the interpolar molecules and they start on the middle on the inside. And then these guys walk to the plus end, these guys walk to the minus end, minus ends pull, plus ends push. Does anyone have any questions? You know, I'm kind of talking a little fast. Oh, I can't see this chat. Is EG5 used for anaphase separation? Yes, I think I talked about that. Oh my God. So yeah, so EG5, yes, because this whole push-pull mechanism is technically part of the forces that pull the chromosomes apart during anaphase. When we talk about anaphase with like anaphase A and B, which will probably be in the review session on Friday. I know I'm pretty sure Sayed's going to go a little more in detail about EG5 with anaphase, but this overall push-pull force is the like driving force for pulling the chromosomes apart during anaphase. So I would say technically yes. I don't know if she's that specific about it though on her like questions, but so yeah, EG5 is basically this. I know she showed them these pictures in class, but this is without monostrol, which is an EG5 inhibitor. So basically what monostrol does is it inhibits EG5. So that pushing mechanism done by EG5 won't occur, but you still have this inward pulling force by the dynein motor proteins for the minus end. So you have this pulling force, but you don't have the opposing pushing force. So as a result, you can see in this picture right here and see the spindle collapses. And can anyone tell me why that would be a very big problem if the spindle collapses? Kind of a
If the spindles collapse, then like they're not going to be able to properly align at the metaphase plate, and thus if they're not properly aligned for that, then they're not going to be able to separate into different cells, and therefore cell division is not going to be able to occur with the amount of equal chromosomes in either cell.
Yeah, perfect. Yeah, definitely. So yeah, your spindle collapses. You can't bind to your chromosomes. You can't pull them apart. The whole thing gets messed up. Yeah, perfect. Couldn't have said it better myself. All right. So now we're at this checkpoint. So the spindle assembly checkpoint is the metaphase checkpoint. So now we're going to go from metaphase to anaphase. This is where the cells make that decision to go from metaphase to anaphase. The checkpoint is activated when the chromosomes aren't lined up correctly. Which is important because if we went back to, where was it? No, sorry, wait, I'm just trying to, here. So if they're not lined up correctly, you won't get this proper segregation, which can lead to a multitude of mutations, which is bad, or program cell death if it doesn't think it can survive. Oh, it's gonna bug me that I didn't make this the same color font, but whatever. Okay, so checkpoint is activated when they're not lined up correctly. So say, I don't know, say you get cohesin doesn't work, they don't stay together and you're all screwed up. So then these two proteins, MAD and BUB, I kind of like to think of them as a duo. They come in, they sense this tension in the pairs. And when they do, this causes CDC20 to be released. So a lot of times in past semesters, students have mixed up CDC20 with CDC25. CDC25 is what activates the M-CDK complex and takes off that inhibitory phosphate that we1 put on that CDK complex. CDC20 is associated with the spindle assembly checkpoint, which is very important for the cell to move from metaphase to anaphase in order for the cells to actually divide properly. So CDC20 is released from a protein called sequestrin and binds to this APC-C complex, which is also shortened to APC. It's just APC. APC is a ubiquitin ligase, which puts ubiquitin on proteins. And as you guys have learned, once you get that ubiquitin tag, the protein will be sent to the proteasome and is distributed. So now you have this CDC20 APC complex, which ubiquitinates securin, which is an inhibitory protein, or securin, sorry, whatever. Securin's job, basically, when it's not, like when it's active and not being ubiquitinated by this complex, it's inhibiting separase, which is another protein. Separase's job is to cleave the cohesin rings that are holding the sister chromatids together, which is important because we eventually will need those sister chromatids to separate, as they will do in anaphase. But we don't want it to cleave the cohesin rings too soon either, because you want them to be aligned right for the perfect timing until you need to separate them. So up until that point, securin's job is pretty important because it's inhibiting separase from cleaving the cohesin rings too soon. But once the cell realizes that it's all set, it's ready to move on to the next step, securin isn't needed anymore. So CDC20 and APC send securin to the proteasome by tagging it with ubiquitin. And now separase can cleave cohesin rings that hold the chromatids together. And then that's when we enter anaphase. So this is this diagram. I know she showed it in class, but you have securin, which is inhibitory, which is inhibiting separase. So then you get this active APC complex, which is activated now after CDC binds to it. It tags securin with ubiquitin. Ubiquitin's degraded. You get this active separase, which cleaves the cohesin rings. Cohesin rings dissociate, and now in anaphase, the sister chromatids can separate. I went through that wicked fast, but that's all I had for you guys tonight. I know Friday they'll probably go over more in detail about anaphase A and B and the anaphase checkpoints and cytokinesis, but that's basically, I know she's ending this week's material with cytokinesis, which is basically just the splitting of the cells. So there's really, if you guys understand up until this point, you've got a good handle on the cell cycle and the processes that regulate it. And I'm sure you're all going to do great on the exam next week, but if you guys have any questions, feel free to ask, stick around, whatever. If you want to go through something more in detail, I'm happy to stay around and help you out. So there's also going to be an exam review next Wednesday. I think Jahan and May are running it. Sad Jason and I aren't running it, but sure, I know Jahan and May will do a great job. But yeah, so if you guys have any questions, feel free to ask. If not, thank you for coming. I'm sorry I went through that pretty fast. But yeah, no problem.
I have a quick question.
Sure.
So it's actually on the problem set.
Yeah.
And I was going through it with Michelle today.
Mhm.
And she and I were like both a little confused on how to properly answer question 3. And I can share my screen if that helps.
Yeah, that actually would work.
Yeah, no
I'll
problem.
share.
Hang on, disabled attendee screen sharing. So I don't know if I can share.
Oh, wait, I can actually, let me share because I can just pull it up.
Perfect, that works.
I'm gonna stop the